any other solute molecule you can think of. And so just like the water stored behind a dam, the water wants to flow downhill, but it can't because it's blocked by the dam. These hydrogen ions, they want to diffuse from their high concentration side to their low concentration side, but they can't because the membrane's blocking them. So it is potential energy. It's kinetic energy waiting to happen. So let's see. The cell membranes block the solutes, and therefore anytime you have a concentration gradient blocked by a membrane or a concentration gradient across a membrane like this, that is potential energy. That's water behind the dam for the cell. Okay, now when we put water behind a dam, that's such a cool picture. Does anybody know where that is? I think it might be Hoover Dam, which is at the Arizona-Nevada border, but I couldn't swear to it. But it is Hoover Dam? Okay, all right. Anyway, so if you ever get a chance, go out and see Hoover Dam. It's amazing. Like these things are buses that are driving across the top of it. It's just this gigantic thing. Anyway, so when we put water behind a dam, we're not doing it just for fun. We're just to store up the energy. We do it because we want to be able to get energy out of it when we need energy. And that's also the philosophy of the cell. It can store potential energy by having a concentration gradient across a membrane, but it usually does that because it wants to eventually get energy out of it. Now, when we want to start getting energy out of the dam, we have to have some sort of spillway, some sort of tube or hole in the dam where we can allow the water to flow through in a controlled way. And for the cell, remember those tubes through the membrane have a name. They're called transport proteins. So these transport proteins are proteins in the cell membrane that essentially form a tube or a gateway through the cell membrane that can allow solutes to pass in a controlled manner. Most textbooks call them channel proteins, or sometimes carrier proteins. Remember that transport proteins are not just a wide open tunnel. They have a binding site, and that makes them specific for just the particular solute that they're supposed to be transporting. If this transport protein was for hydrogen ions, that was really the only thing it would transport because nothing else would fit exactly into its binding site. All right, so let's say it wants to let some of these hydrogen ions on through. One of them would bind right there, and then there's a little flexing of the protein, and it's allowed to pass through. And then maybe another one comes along, gets in the binding site, the protein would flex again and allow it to flow through. Notice that this is diffusion because the solutes are moving from a high concentration area to a lower concentration area. And so this is a spontaneous process. The solutes are losing potential energy because they gain kinetic energy as they zip through the dam to the lower concentration side. And this process you see here has a name. It's called passive transport, or sometimes facilitated diffusion. Both names make sense. Passive because the cell does not have to put any energy into doing it. Solutes diffuse automatically. That's their natural direction, like water going downhill. And the facilitated diffusion also makes sense because it is diffusion, but it has to be facilitated by a transport protein because the diffusion just won't happen without something to allow the solute to pass through the membrane. Spontaneous diffusion of solutes across a membrane, down their concentration gradient of a transport protein. Now, as I was saying earlier, the cell usually doesn't do this just for kicks. It does it because it wants to get energy out of it. And in our real life electrical dams, we have, I don't know, turbines or dynamos, whatever they're called. So as the water rushes down with all that kinetic energy, it turns the electrical generator and you get electricity out of it. Well, cells don't use electrical energy, but they use ATP energy. And so you essentially see the same thing. So ATP is a very high potential energy molecule, and the starting materials to build an ATP molecule are the adenosine diphosphate and the inorganic phosphate. And so what we have to do is the cell has to take that inorganic phosphate and link it back on to the ADP to make an adenosine triphosphate. But that's a very non-spontaneous reaction. That's because that's low potential energy and that's high potential energy. To sort of give you a feeling for it, if this was an ADP and this was an inorganic phosphate, it takes energy to click them back together and make the ATP. If you could grab a phosphate and an ADP and try to click them back up, it'd be like trying to force, I don't know, two magnets that were pointed opposite each other together. It's non-spontaneous. It just won't happen on its own because you're fighting a force to do it. So to get the energy to link those two back together, since it's a non-spontaneous reaction, you have to have an energy source to do it. And that's what the kinetic energy of the solutes flowing through provides. As those solutes, the hydrogen ions, come barreling through, the cell is going to use some of their kinetic energy as the force to click the ATP back together. So that protein can grab hold of an ADP and a PI, and then when that hydrogen goes through there, its kinetic energy provides the energy to click them back together and form an ATP molecule. The transport protein that does that, the transport protein that makes ATPs from ADP and PI as solutes go barreling through, has a name. It's called ATP synthase. I guess I didn't write the full name there, but the process is ATP synthesis. But remember, enzymes usually end in ASE, so the protein is called ATP synthase. That's what you're seeing there on the diagram. Yeah.
Would that non-spontaneous reaction also be an anabolic reaction since you're making something bigger?
That's a good point. Yes, I guess it would count as an anabolic reaction. Excellent. All right. So passive transport is where the solutes come through the transport membrane, and the solutes are losing energy. They're going from their high concentration side to their low concentration side. Now we're going to look at the opposite type of movement through the cell, through the transport membrane, called active transport. And it's essentially movement in the other direction, where the solute is going against its concentration gradient from low concentration side to high concentration side. All right, so let's say the cell, for whatever reason, has a, stop that, there we go. Amazing stuff. Here we go, okay. So let's say the cell has a hydrogen ion on the low concentration side, and it wants to move it to the high concentration side up there on the other side of the membrane. Well, that's a non-spontaneous reaction because the natural tendency of diffusion is to go from high concentration side to low concentration side. So even if we have just a regular old membrane transport protein here, that hydrogen's not going to move against its concentration gradient. Just the opposite would happen. Those ones at the top would come flowing in. But the cell can have some specialized types of transport proteins that can move solutes against their concentration gradient, but it's a non-spontaneous reaction because, well, the solute is gaining potential energy as it goes from low concentration side to high concentration side. So that means it's not going to happen without energy input. And the name of that type of process is active transport, or some people call it pumps. Some people say this is a pumping protein in the cell. Non-spontaneous movement of the solutes across the membrane against their concentration gradient. And it's always going to require an energy source to happen. One of the common energy sources, as you might guess, is ATP, since this is a protein carrying out the work, and pretty much proteins are always powered by ATP molecules. Therefore, typically what will happen is this. Not that. That won't happen. That's like water flowing uphill. Here's what I meant. Oftentimes what happens is this. The pumping protein, the active transport protein, has a binding site for ATP, and then as the solute molecule that's being pumped binds, the ATP molecule, boing, splits off into an ADP and a PI, and the energy of that spontaneous reaction is what provides the kick to move the solute against its concentration gradient. And that's probably the most typical type of active transport protein, is ones that use ATP as their energy source to pump the solutes. But kind of interestingly, there are some active transport proteins that run on electricity, which I don't know, I think it's kind of mind-boggling that we have essentially electrical pumps inside our cells. Now, electricity, I guess I mentioned earlier in the semester, is a flow of electrons. What's actually moving through this electrical cable right here to provide energy for the projector is a flow of electrons. And more specifically, you say that they are high voltage electrons, which you can think of as energized electrons. Well, some of these active transport proteins use a flow of electrons that's moving through some molecules in the cell membrane that carry electrons as the energy source. So floating around within the cell membrane are these groups of molecules that can carry electrons, just like a wire can carry the electrons. And these groups of molecules are called an electron transport system.